So thanks, Equalize, for the program, for the great program, and thank you all for the opportunity to tell you about Clarigene's story. Clarigene is an early stage startup company that spun out in late 2019 from Duke. Our company is committed to bring gene-targeted epigenome therapy for unmet medical need in the CNS disorder, with a focus on age-related neurodegenerative diseases. Clarigene has a well-rounded leadership team. I am a co-founder at Clarigene and also hold a primary position at Duke as a professor and chief at the Division of Translational Brain Sciences in Duke Neurology and Pathology. I have 23 years experience working on the genetic of age-related neurodegenerative diseases, and I've been consecutively funded by federal foundation and pharmaceutical industry grant. My co-founder, Dr. Cantor, is an expert in gene therapy platform and the viral delivery system. And we recently welcomed Elaine Ham, who leads the business development side, and I can only thank Equalize for pairing me with Elaine. So we all recognize that Alzheimer's disease is the sixth leading cause of death in the US and the most common cause of dementia in aging. With the growing aging population, the number of Alzheimer's cases is increasing fast, projected to rise drastically over the next 3 decades, from over 6 million cases in the US alone now to 14 million cases by 2050. The majority of the clinical study target beta amyloid, and those have been failed. So currently, there is no cure for Alzheimer's, no any disease-modifying therapy to slow down the progression of the disease or even to delay its onset. The main challenge is that no treatment addresses the actual cause of the disease. So there is an urgent need to refocus on other targets than the beta amyloid and to shift the paradigm of Alzheimer's drug development toward precision medicine. For this, we first need to understand, why do some people develop Alzheimer's and others do not? Alzheimer's is caused by an interplay between environmental and genetic risk factors, with aging playing a major role. The heritability is large and estimated between 60 to 80%. However, the underlying genetics of Alzheimer's is still far from being fully understood. What we do know to date about Alzheimer's genetics is that the ApoE gene is the leading and the only well-established high-risk factor, whereas the ApoE4 variant is the bad guy. You can picture a city skyline where the ApoE is the tallest skyscraper. I have dedicated the past 15 years since I assumed the faculty position at Duke to investigate the ApoE gene and its mechanistic role in Alzheimer's disease. My lab at Duke studied the ApoE genetic architecture and epigenetic landscape and their crosstalk in Alzheimer's disease. Work from my lab shows that Alzheimer's brain have significant higher level of ApoE than age and sex match control in the analysis of brain structures that are relevant and affected in the disease, as well as in our very, very new data from single cell transcriptomic studies. Other team working on animal model reported that lowering ApoE level in 50% resulted in beneficial effect with respect to the rescue of Alzheimer's pathological changes. That's tuning down ApoE level is expected to have beneficial therapeutic impact for Alzheimer's disease. Clarigene's offer gene therapy approach to target ApoE specifically as an emerging promising target for Alzheimer's disease. Clarigene takes two strategies in developing ApoE targeted therapy, but going forward for today, I will focus on the E4 specific repression strategy. So Clarigene uses CRISPR-Cas technology to develop an IP protected epigenome therapy platform that specifically and precisely repress the E4 allele of the ApoE gene. The platform comprised from all-in-one epigenome editing vector. And how does it work? The concept is built on a single nucleotide difference that distinguishes the risk ApoE form from the natural E3 allele. Using this approach, we are able to bring the platform specifically to the ApoE4 gene variant. And the platform is closing specifically the chromatin at this target site, leading to repression of the ApoE4 variant expression. So the next slide shows an in vitro validation in human iPSC-derived neuron from an individual a case of Alzheimer's disease that have the ApoE4 genotype. You can see here a range of repression of the E4 allele expression that is led by different guides RNA. In this slide, we show an in vivo proof of concept of the platform efficacy in the hippocampus, and we choose the hippocampus as a brain structure that is primarily affected in Alzheimer's disease. In this experiment, we perform a bilateral stereotaxic injection of the reporter gene directly into the hippocampus, and at the same surgery, the left hemisphere of the hippocampus was injected with the epigenome editing vector, and the right hemisphere of the hippocampus with a control vector with no repressor. And you can see a robust and precise reduction in the expression of the reporter gene at the left side compared to the very bright signals that still remain at the right side of the hippocampus. So that is what we have so far. So what is next for Clarigene? So next, we are seeking for a 6 million seed round to complete in vivo proof of concept that will include efficacy, safety, target engagement, dose response, and route of administration studies. Then the following round of funding will be to get us to IND, including the manufacturers of the vector. So who else is in the space as it relates to ApoE targeted therapy for Alzheimer's disease? So this slide summarizes other Alzheimer's therapeutic approaches that target directly the ApoE. It is a very, very busy slide, but the take-home message is that Clarigene is different compared to others by first starting upstream at the production step of ApoE, and also allowing fine-tuning of ApoE expression. We are not doing any ectopic expression or any robust knockout. So this slide shows the pipelines of Clarigene, and I highlighted the lead project for the Alzheimer's program. But the platform has also a potential for other CNS diseases, specifically those that caused by rare dominant mutations. The last slide I would like to summarize. So why to choose Clarigene? We have a novel, efficient platform for CNS epigenome therapy paired with a viral delivery system. The platform is applicable and compatible to other CNS disorders, including rare diseases. We have a scientific founder with deep expertise to advance the company. We have a robust intellectual property portfolio. ApoE targeted system is beneficial for other indications as well. And in addition, the platform has been validated for Parkinson's treatment in vitro and in vivo in a pilot study via our partnership with the pharmaceutical industry. These proof of concept studies for Parkinson are showing no safety issue, and we were also able to achieve the desirable efficacy. So I will conclude by this, and thank you.
Thank you so much, Ornit. We'll go to our Meghana for our first question.
Yeah, no, thanks. That was really interesting. Can you just provide me a little bit more background on your kind of general hypothesis that, you know, lowering ApoE prior to plaque deposition is going to help sort of alleviate and have these preventive protective effects. Can you comment on the kind of the prior data out there that supports this hypothesis?
Yes, sure. So it's based on work from our lab and also work in the field that's showing that ApoE is highly expressed in Alzheimer's brain when we compare it to normal aging individuals. This had been done in study of brain, in autopsy brain. And also now we're conducting single cell analysis, transcriptomic analysis, and we are able to show the specific cells where ApoE is highly expressed in Alzheimer's patients compared to control. There are also animal models that showing where we are targeting ApoE and reducing the expression of ApoE, we can basically rescue the pathology of related to Alzheimer's disease in these animal models.
And then one other question: what endpoints are you going to use for your in vivo proof of concept to test efficacy?
So we will start with pathology based on the characterization of the mice model. We will look for beta amyloid pathology, tau pathology, phosphorylated tau, rescue of loss of neurons. We will also look at plasma and CSF biomarker. Specifically, we will focus on phosphorylated tau forms.
Oh, thank you.
Great, thank you. Chris Borner.
Hey, nice presentation. Just a couple of quick questions. ApoE was, I think, first identified in like the 1970s. So we've known about this one for a long time. Can you characterize sort of what we've learned from any clinical trials that have targeted this approach, and how you're thinking just generally about where others have approached this particular target, what some of the challenges have been and how you overcome them with your planned development approach?
So currently, ApoE is a new emerging target for Alzheimer's disease. And there are no clinical studies, there are preclinical studies. And the table, let me just go backwards so that I didn't went through in details, basically summarize other approaches to target ApoE that are currently in this space. But we are looking at the ApoE expression at the very upstream before it's produced. We are basically modulating the production of the ApoE, and specifically the risk allele, the ApoE4. We are not letting it to produce to a high level.
Thank you.
Great, thanks. Chris Garabedian.
Yeah, sure. So first, a really nice job identifying the target and why it's relevant in Alzheimer's, and kind of why you're excited about this. This is clearly a high risk, high reward disease area, and particularly thinking about a gene therapy that is more preventive and how that might be adopted. And I think there will be a lot of questions around, and Meghana started with this on the how you're going to do optimally the preclinical studies that would be predictive. You have a comparison versus aducanumab. Is that the best, positive control? But so if you want to comment a little bit more on that, that design of the preclinical, but more importantly, the clinical strategy is going to be of great interest to investors. And how are you thinking about getting to the right initial enriched patient population, for that clinical study?
Yeah, I have a slide ready.
Okay.
Okay, so so in order to identify, let me start, I think, with your last question, to identify the patient population, we will start very early. There are before a clinical symptom arise. So we will identify the target population, but first based on genotype, the patients that with the people that are carrying the ApoE4 allele and have family history, and we will measure their plasma and CSF biomarker that were shown to be indicative of to predict or to project the risk to develop Alzheimer's disease.
Great. Yeah, thank you. And you know, I know we don't have time to cover it now, but the elucidating more of the competitive landscape and their strategies and how you might learn from their approaches and maybe differentiate yourself. But overall, nice presentation. Thank you.
Thank you.